One of the outputs of this workflow is the consensus sequences for all samples. Essentially, what the workflow does, it calls variants, and then it looks at the reference, changes the nucleotides corresponding to variable sites in the reference to that allele, and prints it as a sequence for a given sample, and does it for all samples. So essentially, it generates data set. Well, that looks like this. So these are the sequences of measles genome corresponding to individual samples in our data with the substitutions put in there. We can use this data set to see if there's anything interesting going on with the substitutions in terms of, for example, do any of them look like these are adaptive changes? Perhaps viruses evolving towards some goal, some higher fitness goal in a sense. So in order to do that, we need to take these consensus sequences, splice them by genes, so meaning extract from the whole genome alignments corresponding just to the genes of interest, and then analyze them using a tool which detects positive selection. So two steps here. First, we take the sequences, slice them by genes, and we put these gene sequences back into Galaxy history, and then we run a tool which is designed specifically for identify sites under selection. The slicing we're going to do using a Jupyter notebook as well, because again, it's a complicated case with these overlaps. So we need to, it requires a bit of a custom code. I prepared another notebook here. And again, make sure that this is on. So let's start this notebook. Okay, just like before, what this notebook does, it needs to read the GenBank file. So let's see, our GenBank file is 1598. And our multiple sequence alignment or sequence file is 1366. Okay. Let's just again start kernel just like we did before and run all cells. Again, this is done using ChatGPT. So the first step here is, well, actually the only step here really, it filters sequences by length just to ensure that, just for sanity, that all sequences in this file have the same length. And then it reads the GenBank file and slices them by genes. And that slicing by genes generates these files. So it does this for all genes, but we're not interested in all genes. We just need really the three reading frames. So the phosphoprotein, protein C, and protein V. And the way we put data back into Galaxy is by using this interesting expression. So essentially, we want this file here. Let's concentrate on the V protein for now. The paper describes all of them, but in this example, let's just do V. So I'm gonna insert this here, call it V, and just run that cell. I guess when we executed the cell, the P was already there. Let me delete this because this. Let's do V for now, just V. So you can see using this command, it puts data from Jupyter into Galaxy. And that's a multiple alignment file. Oh, good. The tool that I'm going to use to detect sites under positive selection is called MEME. One of the it actually takes two inputs. It takes the multiple alignment, but it also needs a guide tree. So it needs a phylogenetic tree. So let's quickly build that tree. I'm going to go to Tools and look for the RapidNJ. It's a tool which rapidly generates trees, rapidly generates neighbor joining trees. So we want to do this for this file V. This is a DNA sequence. And let's keep everything the same. Let's just run it. Okay, let's look at this tree. So it's a tree. You can see how our samples stratified in actual studies. Because if we, for example, cover them by location, say Romania versus US versus Canada, we will see that stratification. But we don't need to do this right now. So the next, I'm going to run meme to run this tool. It's hi meme. So it needs the alignments. It needs a phylogenetic tree. We are going to do this for internal branches only because we want to avoid errors. For example, we want to avoid sites which are changed only in one sample. These are not reliable. So we are just going to examine sites which appear essentially in more than one sample. Let's run it. So results are done, and let's look at this. So we can visualize results of hi-fi. And if we scroll down and visualize this as the plot for p-values, that's actually the one we need, we will see that we have two sites with the smallest p-values, 105 and 111. And these are the sites identified by meme as likely under diversifying selection. So this is how we can quickly go from essentially alignments to conclusions such as this.